The third type of signaling pathway is called the receptor tyrosine kinase pathway, or RTKs. Now, your book refers to them as enzyme-coupled receptors, but the rest of the world refers to them as receptor tyrosine kinases, and so that's what we'll use going forward. Receptor tyrosine kinases are of intense interest to the pharmaceutical industry, since these are the types of pathways that are most often involved in promoting cellular growth. And aberrant cell growth is one of the hallmarks of cancer. So the thought is if you can get drugs to interfere with these pathways that are promoting cell growth, you might have a potential treatment for cancer. Receptor tyrosine kinases exist in a monomeric inactive form. They bind to a ligand, which is always a dimer or has a dimeric binding moiety. This enables two monomers to become close to each other. This activates the receptor. The receptor gets activated because both of the cytoplasmic tails can possess kinase activity, and the first thing they will do is phosphorylate each other. This now activates this cytoplasmic tail further, allowing other proteins to bind, and they will get phosphorylated by the kinases on the cytoplasmic tail, and then you can proceed with the downstream cascade of phosphorylation events that transmit the information. These pathways tend to rely on phosphorylated tyrosines. As we've mentioned, there are three amino acids that can be phosphorylated: serine, threonine, and tyrosine. Serines and threonines are phosphorylated much more often than tyrosines. Tyrosines, which are relatively rare, account for only about 2% of the total amount of protein phosphorylation in cells, and they are fairly specific to these growth-promoting pathways. So that's why receptor tyrosine kinase inhibitors, or even just tyrosine kinase inhibitors, TKIs, are most often used almost exclusively to treat different cancers. This slide sort of highlights the manner in which the receptors are activated. It's almost hard to imagine that this was a poorly understood and much puzzled about phenomenon in biology for a very long time. The conceptual difficulty was that receptor tyrosine kinases are known to possess a single alpha helix that crosses the membrane. And so the question was, how can a binding event, which occurs on the exoplasmic side, produce a conformational change which can be transmitted through just the single alpha helix to produce a change here? And it turns out now we know the answer is so much simpler. It's that the ligand molecules are dimeric, and it's not necessarily a conformational change promoted vertically through the protein, but in the fact of bringing two receptors closer together so that they can phosphorylate each other. So this is an important concept. It's referred to as ligand-induced dimerization. Ligand-induced dimerization. As I've mentioned, receptor tyrosine kinase pathways often stimulate cell growth, and one of the leading players in promoting cell growth is this protein called RAS. RAS is a lipid-bound, so constrained to the membrane, surface of the membrane, monomeric GTPase. So when it's bound to GTP, it is active. It will transmit the signal via phosphorylation of downstream proteins. It's a GTPase, so after a certain amount of time, it will hydrolyze the GTP, producing GTP, and this is the inactive form of RAS. RAS is initially activated, again, by ligand-induced dimerization, activation of the receptor, activation of a RAS activating protein, which serves as a nucleotide exchange factor, promoting factor. It promotes the removal of GDP and the binding of GTP to RAS, which is what activates it. Here we once encountered the MAPK module of signaling intermediate proteins. Now we're seeing them in higher eukaryotes. We saw them earlier in yeast when they were involved in osmolarity sensing. Individual MAP kinase proteins in humans are sometimes referred to as RAF, MEK, and ERK, and you might consider that an improvement over the generic names, which are MAP kinase, 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 and so on. But again, the way it goes is RAS is a kinase, it will phosphorylate the MAP kinase. This protein RAF hydrolyzes ATP, uses that extra phosphoryl group to attach it covalently to the protein. Now this enzyme is activated. It will hydrolyze ATP, take another phosphoryl group, attach it covalently to its target protein, in this case the MAP kinase, which can then hydrolyze ATP and phosphorylate another proteins, including transcription factors, which will transcribe new genes, or proteins which can change their activity. Now I want to remind you that these are enzymes, so enzymatic processes, so they're capable of signal amplification. A single enzyme here can phosphorylate many MAP kinase proteins. And similarly, MEK can phosphorylate many copies of ERK. So I don't want you to think that this is a phosphate shuttle where this phosphate is being then transferred to this protein. That's not the case. These phosphates stay on this protein. New phosphates are obtained from ATP, which is the phosphorylation of the downstream proteins. This pathway will be inactivated here when phosphatases come and cleave away the phosphate groups. Mutated RAS is one of the leading causes of cancer. When RAS is mutated, it loses its ability to hydrolyze GTP. So GTP is stuck to the protein is stuck on the on position, and it's going to phosphorylate the MAPK proteins even though it's not supposed to. There's no growth ligand out here. There's no dimerization of the receptor. RAS should be turned off. Unfortunately, the mutation prevents it from being turned off. And again, RAS was one of the first known causes of cancer, and statistically it's one of the most frequent. Unfortunately, as far as I know, there are not good therapies to interfere or block an aberrantly activated RAS protein. We conclude with a slide that shows the interactions among many different signaling pathways. You will encounter diagrams like this frequently in biology, but I'm personally always concerned that they frequently result from an excessive zeal of arrow drawing. There's no question that signaling pathways interact, but it seems to me that it often happens that once a paper appears in the published literature and a pathway becomes established, another paper will come along and they'll add a few more arrows, and so will the next paper. And after tens of thousands of papers about signaling pathways, many arrows accumulate. I would say that it's a very few minority of papers that have the conviction to prune away arrows that have been drawn previously. What these slides generally reveal to me is that we're still at a very early stage in understanding the important connectivity between signaling proteins. And there's still a lot to learn about how cells maintain the fidelity and the specificity of their signals. It may well be that the truth is often quite complex and involving a lot of proteins and arrows, but a full and accurate knowledge of wiring within the cells is very much still an area of research for the future. Okay, thanks for listening.